Here is a great, deep, and detailed summary of the passage, written in the most simple, clear, and beginner-friendly way possible, with extra explanation and examples to help you understand every part. Deep, detailed summary. Are living things and non-living things made of the same materials? Yes, but there is a big difference in how much of it they contain. 1. Things on Earth. Earth is full of life. Animals, plants, bacteria, fungi, and more. All of them together form what we call the biosphere, the part of the Earth where life exists: land, water, and air. Even though living things look very different, scientists ask, are all living things made of the same basic substances? 2. Everything is compounds. Everything in the world, living or not, is made of chemicals. Elements are basic substances like carbon, oxygen, and hydrogen. Example, carbon, C, oxygen, O, hydrogen, H. Compounds are made when elements combine. Example, water equals H2O, hydrogens, 3. Scientists use elemental analysis to check what something is made of. Scientists can test a plant, an animal, or microbes, tiny organisms, and find out which elements are inside and how much of each. This tells us the chemical makeup of a living thing. Example, if we test a piece of leaf, we might find 40% carbon, 30% oxygen, 10% hydrogen, 5% nitrogen, and so on. 4. Non-living things like rocks and soil also have the same elements. Scientists do the same test on non-living things, like a rock or sand. They also find carbon, oxygen, hydrogen, etc. So both living and non-living matter are made of the same types of elements. 5. So what is the difference? If we just look at what elements are present, there is no major difference. The same elements are found in rocks and in living things. 6. But the difference is in the amounts, how much of each element is present. When we look closer, we see that living things have a lot more carbon and hydrogen. Non-living things like rocks have more silicon and oxygen. For example, element in living things in Earth's crust, rock, silicon, Sc, high 40%, very low 0.03%. Hydrogen H, high 10%, low 0.14%. Oxygen O, present 30%, very high. Silicon S, very low 0.28%. 7. Why is this important? It tells us that living things are rich in certain elements, especially carbon, the backbone of life, found in all proteins, sugars, DNA, etc. Hydrogen, found in water, fats, and more. These elements help build cells and life structures. So even though living and non-living things share the same elements, the pattern and amount of these elements make life unique. Final simple summary. 1. Living things and non-living things are made of the same basic elements. 2. But living things have a much larger amount of carbon and hydrogen. 3. This chemical difference helps us understand what makes living things special. 4. So what makes something alive is not new elements. It's the special use and arrangement of common elements like carbon and hydrogen. Would you like me to now give you a comic strip version of this with cute visual examples of living and non-living? Absolutely. Here's a great, deep, and detailed summary of the entire passage, written in the most simple, clear, and easy to understand way for total beginners, with extra explanation and real-life examples to make everything 100% crystal clear. Deep, simple summary. How to analyze chemical composition in living organisms. What is the main goal? Scientists want to know what chemicals are inside living things. What kinds of substances, like sugars, proteins, vitamins, etc., make up a plant, animal, or microbe? Why do we want to know this? To understand how living things work, to study what makes life possible, to learn about health, disease, growth, and energy. How do scientists find these chemicals? They use a method called chemical analysis, which means testing and breaking down a living thing to see what it's made of chemically. Step-by-step process. Step 1. Take a sample of a living tissue. This could be a like a piece of carrot, a leaf, a piece of liver from an animal, a paste full of microbes, tiny living creatures. These tissues are full of cells, and cells are full of important chemicals. Step 2. Crush the tissue in acid. Scientists use a chemical called trichloroacetic acid, ClCCOOH. They grind the tissue using a mortar and pestle, bowl and stick. Helps to break open the cells, release the chemicals inside. Example, it's like crushing garlic to release its juice. We crush the tissue to release the cell juice. Result, a thick paste called a slurry. Step 3. Strain the slurry through a cloth. Scientists pour the thick slurry through cheesecloth or cotton. This separates it into two parts. 1. Liquid part, called the filtrate or acid-soluble pool. It contains small, important organic molecules that dissolve in acid. Examples, sugars, like glucose, which gives energy, amino acids, which build proteins, nucleotides, which build DNA and RNA, vitamins, fatty acids, parts of fats. This part is very useful because it has the key building blocks of life. 2. Solid part, called the retentate or acid-insoluble fraction. It contains big, non-dissolved particles like cell walls, big proteins, cell parts like the nucleus or mitochondria. Important, but they are too big to dissolve in acid, so they stay on the cloth. Example to help you picture it. Think of making orange juice. You crush an orange, like grinding tissue. You strain it through a strainer, like using cheesecloth. You get juice equals liquid with vitamins and sugar, acid-soluble pool. Pulp, thick bits left behind, acid-insoluble part. It's the same idea in science, but with living tissues and acid. What do scientists learn? From the acid-soluble part, scientists have discovered thousands of organic compounds. These compounds tell us how life works. They are used to study metabolism, nutrition, disease, and genetics. Final summary, super simple. 1. All living things are made of tiny chemical compounds. 2. To study them, we take a small piece of living tissue, crush it in a special acid, strain it through a cloth. 3. We get two parts. A liquid part, acid-soluble pool, has small, important molecules like sugars and amino acids. A solid part, acid-insoluble, has large particles like cell parts. 4. Scientists study the liquid part to understand what chemicals are inside living things. 5. These chemicals are the basic building blocks of life. Why is this important? This method helps us understand the chemistry of life, know what makes up our cells and bodies, study how food, energy, and growth work inside us, learn how to treat diseases, and improve health. Would you like me to now give you an easy chart of the types of compounds found in the acid-soluble part, like proteins, sugars, fats, and more? Absolutely. Here's a great, deep, and detailed summary of the entire passage in the simplest possible language, just like a brilliant teacher would explain it to a complete beginner. Summary. How do we study the chemicals inside living things? Main idea. Living organisms like plants, animals, and microbes are made up of many kinds of chemicals. Scientists want to understand what those chemicals are and what role they play in life. To do this, they perform chemical analysis on living tissues. Step-by-step summary of the process. 1. We can find both organic and inorganic compounds in living things. Organic compounds. These are carbon-based molecules like sugars, fats, proteins, DNA. We call them biomolecules because they are found in living things. Inorganic compounds. These are non-carbon substances like water, minerals, calcium, iron, phosphates, sulfates, etc. Living organisms have both types. 2. To study organic compounds, we extract and separate them. Scientists grind the living tissue, like a leaf or piece of liver, using a chemical like trichloroacetic acid. This breaks open the cells and releases the chemicals inside. The liquid mixture, extract, is then filtered to remove solids. The liquid part contains small organic compounds like sugars, amino acids, build proteins, fatty acids, build fats, nucleotides, build DNA, RNA, vitamins. Then, using special separation techniques, scientists isolate one compound at a time and purify it so it can be studied. 3. Once separated, we use tests to learn more. These are called analytical techniques. They tell us the molecular formula, like C-H2OC for glucose, the structure of the molecule, how the atoms are connected. 4. All these carbon-based molecules are called biomolecules. Examples of biomolecules: glucose, a sugar. Amino acids make proteins. Fatty acids make fats. Nucleotides make DNA and RNA. Enzymes, hormones, vitamins. These molecules are very important for life. They do the work inside our cells and give us energy, strength, growth, and reproduction. 5. But living things also have inorganic elements. How do we know that? We perform a different kind of experiment. 6. To study inorganic compounds, we burn the tissue. First, we weigh the tissue when it's fresh. This is called wet weight. Then we dry it to remove water. What's left is dry weight. Next, we burn the tissue completely. This removes all carbon-based parts. They become gases like CO2 and water vapor. The leftover ash contains inorganic elements and salts, like calcium, magnesium, sodium, phosphate, sulfate. So, from the ash, we learn what non-living substances are also part of living things. 7. Inorganic compounds are also found in the extract. Some inorganic salts, like phosphate and sulfate, are also present in the liquid extract from earlier. 8. Types of analysis, elemental versus compound. Elemental analysis tells us what elements are there, like carbon, hydrogen, oxygen, nitrogen, chlorine, calcium, etc. Compound analysis tells us what types of molecules are there, like sugars, proteins, fats, DNA, salts like phosphate and sulfate. 9. Different ways to classify compounds. Chemistry versus biology. From a chemistry point of view, we group molecules by their functional groups, like aldehyde, ketone, alcohol, aromatic ring. From a biology point of view, we classify molecules by what they do in life, like amino acids make protein, fatty acids make fat, nucleotides make DNA, RNA, sugars give energy. Final takeaway. 1. Living organisms are full of chemicals, both organic, carbon-based, and inorganic, non-carbon. 2. We can extract, separate, and study these compounds from tissues. 3. Organic compounds are called biomolecules. They include sugars, fats, proteins, DNA, etc. 4. Inorganic elements are found in ash after burning the tissue. 5. Science uses both chemical structure and biological function to classify these substances. Would you like a simple diagram or table to visually compare? Organic versus inorganic compounds, chemistry versus biology classification. Here is a great, deep, and very detailed summary of the full explanation about amino acids in the simplest possible language, like you're learning for the first time. Deep, detailed summary, amino acids. What are amino acids? Amino acids are small chemical units that join together to form proteins, which are essential parts of all living things, including your skin, muscles, blood, and even enzymes and hormones. They are organic compounds, which means they are made using carbon along with other elements like hydrogen, oxygen, and nitrogen. Basic structure of an amino acid. Each amino acid has a central carbon atom called the alpha carbon, A carbon, which is connected to 4 different groups. 1. Amino group, written as NH2, basic in nature. 2. Carboxyl group, written as CoH, acidic in nature. 3. Hydrogen atom, a single hydrogen H4. 4. R group. This is a side chain, and it is different in each amino acid. The R group is what makes each amino acid unique. Why are they called alpha amino acids? They are called alpha amino acids because both the amino group, mine is an NH2, and the acid group, my COOH, are attached to the same alpha carbon. Amino acids are like modified methane. Methane, CH4, is a simple molecule with full 1 carbon and 4 hydrogens. In amino acids, 3 of those hydrogens are replaced by an amino group, a carboxyl group, an R group. 1 hydrogen stays. So you can think of amino acids as methane with special attachments. R group, the special part. The R group, or side chain, can be small or large, simple or complex, charged or uncharged, acidic, basic, or neutral. Because the R group changes, many kinds of amino acids exist, but only 20 types are commonly used by living things to make proteins. Examples of amino acids and their R groups. Amino acid, R group description, extra hydrogen. Glycine, just a hydrogen H. Alanine, a methyl group, CH3. Serine, a hydroxymethyl, CH2H. Glutamic acid has extra acid group. Lysine has extra amino group. Tyrosine has an aromatic ring. Classification of amino acids based on R groups. 1. Acidic amino acids. R group contains extra carboxylic acid group, a COOH. Example, glutamic acid. 2. Basic amino acids. R group contains extra amino group, example, lysine. 3. Neutral amino acids. R group is neither acidic nor basic, example, valine. 4. Aromatic amino acids. R group contains a ring structure, aromatic ring. Examples, tyrosine, phenylalanine, tryptophan. How amino acids react in different pH. The amino group, NH2OH, and the carboxyl group, COOH, can gain or lose hydrogen ions, H-pies. This makes them ionizable, meaning they can carry positive or negative charges. In acidic or basic solutions, different pH levels, amino acids may change shape and charge. This is very important in biology because different parts of the body, like stomach, blood, and cells have different pH levels, and amino acids must adjust in those environments. What are amino acids used for? Build proteins, like muscles, enzymes, hemoglobin, help in growth and repair of the body, make hormones, like insulin, help in metabolism, used in signaling, immune function, and transport of substances. Final super simple takeaway. Amino acids are the basic building blocks of proteins. They all have the same base structure, but differ in the R group. There are 20 amino acids used to make proteins. Based on their chemical groups, amino acids can be acidic, basic, neutral, or aromatic. They can gain or lose charges depending on the pH of the environment. Their behavior and function are controlled by their 1. Amino group, NH2, 2. Carboxyl group, COOH, 3. R group, varies in each. Would you like a colorful table or flashcards for all 20 amino acids, or a visual diagram of an amino acid labeled with all parts, NH2, COOH, HR? Of course. Here is a great, deeply detailed, and super simple summary of the entire topic on lipids, just like a world-class teacher would explain to a total beginner. This summary covers everything clearly, connects all the parts, and uses easy words, strong visuals, and examples so you can truly understand and remember. Deep, detailed summary of lipids in simplest words. 1. What are lipids? Lipids are a group of natural substances. They include fats, oils, waxes, some vitamins, A, D, E, K, cholesterol, and other body fats. Lipids are very important for giving energy, storing fat, building cell membranes, helping brain and nerves work. Important feature, lipids are water insoluble, meaning they don't mix with water. Like when you put oil in water, it floats. 2. Fatty acids, the building blocks. A fatty acid is a long molecule with a head, called carboxyl group, written as OH, a tail, called R group, made of carbon atoms. Think of a fatty acid like a lizard. Head equals COOH, acidic part, tail, long chain of carbon atoms, R group. The R group can be short, like 1 to 2 carbon atoms, or long, up to 19 carbon atoms. 3. Examples of fatty acids. Palmitic acid, found in ghee butter, has 16 carbon atoms, 1 head plus 15 tail. Arachidonic acid, found in brain meat, has 20 carbon atoms, 1 head plus 19 tail. 4. Saturated versus unsaturated fatty acids. Type, bonds, shape, state at room temp. Example, donic acid. Saturated, single, straight, solid. Butter, ghee. Unsaturated, double. Bent, liquid. Mustard oil, olive oil. Saturated equals no double bonds, packed tightly, solid. Unsaturated equals 1 or more double bonds, loose packing, liquid. 5. Glycerol, the connector. Glycerol is a small, sweet, oily molecule. It has 3 carbon atoms, each with an OH hydroxyl group. It acts like a 3-hook hanger to which fatty acids attach. 6. How are fats made? Fats are made when glycerol joins with fatty acids. This process is called esterification. Water is removed. A chemical bond called ester bond is formed. Based on how many fatty acids are attached to glycerol, name, number of fatty acids, found in esterification. Monoglyceride, 1. Rare processed foods, diglyceride, 2. Food emulsifiers, triglyceride. 3. Main fat in our body and food. Triglyceride equals most common fat. 7. Fats versus oils feature fat, oil, it's fat. State at room temp solid, liquid. Melting point high, low. Example, ghee, butter, vanaspati, mustard oil, coconut oil. In cold weather, ghee becomes hard, it's fat. Mustard oil stays liquid, it's oil. 8. Special lipids. Phospholipids. Some lipids are not for energy, but for structure. These lipids have 2 fatty acids, 1 glycerol, 1 phosphate group, 1 organic group, like choline. These are called phospholipids. 9. Where are phospholipids found? In the cell membrane, the thin outer cover of every cell. Phospholipids form a double layer. Water, tails hide inside, away from water. They help protect the cell and control what goes in and out. 10. Example of a phospholipid, lecithin. Lecithin is a common phospholipid found in egg yolk, soybeans, sunflower seeds, used in food like chocolates, mayo, to mix oil and water. 11. Complex lipids in brain and nerves. Tissues need special lipids. These lipids have complicated structures. They help in memory and brain development, protecting nerves. Example, myelin sheath. Final quick recap. Topic, simple fats and oils that don't mix with water. Fatty acids, long-chain molecules with a COOH head and a carbon tail. Saturated fatty acids, solid fats without double bonds, like butter. Unsaturated fatty acids, liquid oils with double bonds, like mustard oil. Glycerol, 3-carbon base for attaching fatty acids. Triglycerides, main fats formed from 3 fatty acids plus 1 glycerol. Phospholipids, special fats with phosphate found in cell membranes. Lecithin, a common phospholipid used in food and cells. Brain lipids, complex lipids needed for brain and nerve function. If you'd like, I can also give a short quiz to test your memory. Tricks to remember this easily. Diagrams to make it visual. Would you like any of these next?
Of course, here's a great, deep, detailed summary, but still in very simple words, so you can get the big picture and the important details together. 1. Life and carbon compounds. All living things, plants, animals, humans, bacteria, are made of carbon compounds, which are molecules with carbon atoms. These carbon compounds include proteins, sugars, fats, and the genetic material DNA and RNA. 2. Heterocyclic rings. Many important molecules in living things have heterocyclic rings, which are ring-shaped structures made of different atoms. Hetero means different, and cyclic means circle. In DNA and RNA, these rings have carbon and nitrogen atoms. Example, picture a bracelet with black and blue beads, carbon and nitrogen. That's like a heterocyclic ring. 3. Nitrogen bases. The letters of life. There are 5 main nitrogen bases. Adenine, A. Guanine, G. Cytosine, C. Thymine, T, only in DNA. Uracil, U, only in RNA. These bases are like letters of a secret code used by cells to store genetic information. DNA uses A, G, C, T. RNA uses A, G, C, U. U replaces T. NABAS 4 bases combine with sugar to make nucleosides. Each nitrogen base can attach to a sugar molecule. The sugar can be ribose in RNA, deoxyribose in DNA. Deoxy means it lacks an oxygen atom compared to ribose. When a base sticks to a sugar, it forms a nucleoside. Examples of nucleosides: adenine plus sugar, adenosine; guanine plus sugar, guanosine; cytosine plus sugar, cytidine; thymine plus sugar, thymidine; uracil sugar, uridine. 5. Nucleosides add phosphate to make nucleotides. If a phosphate group is also attached to the sugar at the 5' carbon of the sugar, the nucleoside becomes a nucleotide. Nucleotides are like the complete building blocks of DNA and RNA. Examples of nucleotides: adenosine plus phosphate, adenylic acid, AMP; guanosine plus phosphate, guanylic acid, GMP; cytidine's phosphorylic acid, CMP; thymidine plus phosphate, thymidyllic acid, TMP; uridine's phosphate, uridylic acid. Important point: the phosphate group allows nucleotides to connect with each other in chains. 6. Nucleotides join to make nucleic acids. Nucleotides connect together by bonds between the phosphate of one nucleotide and the sugar of the next. This forms long chains, which we call nucleic acids. There are 2 types of nucleic acids: DNA, deoxyribonucleic acid, 7, and RNA, the genetic material. DNA is the permanent library of instructions for how to build and run a living thing. It stays in the cell nucleus in eukaryotes. RNA helps read DNA's instructions and build proteins. It carries the information to the cell's machinery, ribosomes, to make proteins. Together, DNA and RNA are the genetic material, which means they store, copy, and transmit the information needed for life. 8. Summary of how it all connects. 1. Nitrogen bases, A, G, C, T, U, are like letters. 2. Bases plus sugars, nucleosides, letters with handles. 3. Nucleosides phosphate, nucleotides, complete beads with hooks. 4. Nucleotides join nucleic acids, DNA, RNA chains like necklaces. 5. DNA, RNA store and pass on instructions for building everything in a living thing. 9. Why it matters? Aren't just for DNA, RNA. They also help with NTP, adenosine triphosphate, is the main energy tells you currency of cells, and it's a new signaling. Molecules like C-camp are nucleotides used for cell signals. DNA gives instructions for everything from eye color to metabolism. RNA reads DNA's instructions and helps build proteins which do the work in your body. Final deep detailed takeaway. Life's information is stored in DNA and RNA, which are long chains of nucleotides. Nucleotides are made of 3 parts. 1. A nitrogen base, a GCTU. 2. A sugar, ribose or deoxyribose. 3. A phosphate group. These nucleotides connect like beads on a string to make nucleic acids. The order of the bases in DNA, RNA forms a code that tells cells what proteins to make, controlling everything about a living organism. 1-sentence summary. DNA and RNA are chains of nucleotides made of base plus sugar phosphate, and they carry the genetic code that directs all life's processes. Would you like a comic strip, diagram, or mnemonic trick to remember this? Of course, here's a great, deep, detailed summary in the simplest words possible, so you truly understand the whole topic from start to finish without missing any important point. 1. What are metabolites? Metabolites are chemicals made by living things, plants, animals, microbes, during life processes. They come from metabolism, which is all the chemical reactions happening in a living thing to stay alive, grow, and reproduce. Examples of metabolites: sugars, amino acids, pigments, toxins. 2 main types of metabolites. All metabolites made by living organisms can be grouped into 2 main types. Primary metabolites. Definition: chemicals directly needed for basic life processes like growth, energy production, building cells, and reproduction. Found in all living things, plants, animals, bacteria, fungi. Roles: give energy, e.g. glucose, main energy source. Build proteins, e.g. amino acids, make muscles, enzymes. Make nucleic acids, e.g. nucleotides, DNA, RNA. Form cell membranes, e.g. fatty acids, build protective layers of cells. Examples: glucose provides energy, amino acids building blocks of proteins, fatty acids make up fats and membranes, nucleotides build DNA, RNA. Secondary metabolites. Definition: chemicals not directly needed for basic survival but give extra advantages to plants, fungi, or microbes. Found in mostly plants, fungi, bacteria, rarely in animals. Roles: defense, keep insects or animals away, e.g. toxins. Attraction, bring pollinators like bees with bright colors or sweet smells. Competition, kill or slow down other plants or microbes growing nearby. Communication, send signals to other plants or animals. Examples: alkaloids, morphine, painkiller, nicotine in tobacco. Pigments, carotenoids, orange in carrots, anthocyanins, red, purple in berries. Essential oils: lemon oil, eucalyptus oil, scents. Toxins: ricin, abrin, very poisonous. Gums and rubber, materials from plants. Stat 3. Primary vs secondary metabolites. Key differences. Feature: primary metabolites, secondary metabolites. Found in plants and bacteria. Needed for survival? Yes. Essential for life? No, but provides special advantages. Found in all living things, plants, animals, microbes, mostly plants, fungi, bacteria. Function: growth, energy, building basic cell parts. Defense, attraction, competition, communication. Examples: amino acids, glucose, nucleotide, morphine, curcumin, pigments, essential oils. 4. Why do organisms make secondary metabolites? Plants cannot move, so they need other ways to protect themselves, attract helpers, or compete with others. Defense make bad-tasting or toxic chemicals, e.g. alkaloids like nicotine, to keep animals from eating them. Attraction make bright colors or sweet smells to bring bees, butterflies, or birds for pollination. Competition release chemicals into the soil to stop other plants from growing nearby, called a allelopathy. Communication send out scents to warn nearby plants of danger, e.g. being eaten by insects. 5. Why are secondary metabolites important for humans? Even though secondary metabolites aren't needed for the plant's basic survival, they are very important for us. Medicines: morphine, painkiller; vinblastine, anti-cancer drug; curcumin, anti-inflammatory; spices and flavors: black pepper, turmeric, vanilla; scents and perfumes: lavender oil, rose oil; materials: rubber for tires, gloves, dyes and pigments; natural colors for food, fabrics; pesticides: natural toxins that protect crops. 6. Detailed examples of secondary metabolites, table 9.3. Pigments: carotenoids, orange, yellow, carrots; anthocyanins, red, purple, berries; alkaloids: morphine, codeine, pain relief; terpenoids: monoterpenes, diterpenes, resins, essential oils; essential oils: lemongrass oil, scents, insect repellents; toxins: abrin, ricin, extremely poisonous; lectins: concanavalin A, binds to sugars used in labs; drugs: vinblastine, treats cancer; curcumin from turmeric, reduces inflammation; polymeric substances: rubber, tires, gloves; gums, food thickeners; cellulose, plant cell walls; paper, cotton. 7. How do scientists use primary and secondary metabolites? By isolating and studying these chemicals, scientists can understand how life works at the molecular level, discover new drugs for diseases, make natural products for everyday use, perfumes, flavors, dyes, create sustainable materials, biodegradable plastics from plant cellulose, protect crops using natural pesticides. 8. Why is this topic exciting? The world of metabolites shows how creative nature is by making thousands of unique chemicals to help plants, animals, and microbes survive in different environments. Many of the products we use every day come from secondary metabolites discovered in plants or microbes. Scientists continue to explore new metabolites, which might lead to the next big medicine, eco-friendly material, or natural flavor. Super simple. Living things make chemicals. Primary metabolites must have chemicals for life, energy, growth. Secondary metabolites, extra chemicals that help with defense, attraction, competition, and give humans many useful products. Studying these chemicals helps scientists find new medicines, materials, and understand life better. Would you like a simple cartoon or memory trick to remember primary versus secondary metabolites? Absolutely! Here's a great, deep, detailed summary in simple, clear words that brings together every important idea about micromolecules and macromolecules, so you have a complete, connected understanding. What are biomolecules? Biomolecules are the chemicals found in living organisms, in plants, animals, bacteria, fungi, everything alive. They are the building blocks and machinery of life, responsible for energy, structure, information storage, and countless life processes. 2 big categories of biomolecules. Scientists classify biomolecules into 2 main groups based on their size, molecular weight, and whether they dissolve in acid. 1. Micromolecules, small molecules. 2. Macromolecules, biomacromolecules, big molecules. 1. Micromolecules. Definition: small biomolecules with molecular weight less than 1,000 Daltons. Where found? Found in the acid-soluble pool, meaning they dissolve when tissue is treated with acid. Examples: water 8 to 18 day, glucose 180 day, amino acids 75 to 200 day, vitamins 100 to 800 day, mineral ions, sodium Na+, potassium K, calcium Ca². Importance in life: provide quick energy, glucose; serve as building blocks, amino acids for proteins; actors, cofactors, vitamins that help enzymes work; move easily inside cells and participate in fast reactions. Key property. Because they are small, micromolecules can diffuse easily, mix quickly in the cell, and are used rapidly. 2. Macromolecules, biomacromolecules. Definition. Large biomolecules with molecular weight typically above 10,000 daltons, often hundreds of thousands or even millions. Where found? Found in the acid-insoluble fraction, meaning they do not dissolve in acid. They remain as solids when tissues are treated with acid. 4 major types. 1. Proteins, long chains of amino acids, do most of the work in cells, like enzymes, antibodies, transporters. 2. Nucleic acids, DNA and RNA, long chains of nucleotides, store and transfer genetic information. 3. Polysaccharides, long chains of sugars, store energy, starch, glycogen, or provide structure, cellulose in plants. 4. Lipids, large molecules made of fatty acids, form membranes, store energy, act as signals. Importance in life. Proteins catalyze chemical reactions, enzymes, transport molecules, provide structure, keratin in hair. DNA, RNA hold and express genetic instructions. Polysaccharides store fuel, starch, glycogen, build strong structures, cellulose in plant cell walls. Lipids build cell membranes, store long-term energy, send signals between cells, hormones. Key property. Macromolecules are complex, stable and specialized, often with 3D shapes that give them unique functions. Why do scientists use acid solubility to separate them? When scientists crush cells and add acid, micromolecules dissolve. They are in the acid-soluble pool. Macromolecules don't dissolve. They stay in the acid-insoluble fraction. This simple method helps scientists separate and study small and big molecules from tissues. Molecular weight. Difference. Molecular weight measure molecule is using the unit Daltonaire. Micromolecules usually 18 to 800 dar, definitely 1000 dar. Macromolecules 10,000 dar and above, often much larger. Examples for scale: glucose 180 dr micromolecule, a small protein like insulin 6,000 dr small macromolecule, a big protein like hemoglobin 64,000 dr. DNA can be millions of daltons long. Why does this matter? Dividing biomolecules into scientists understand what roles different molecules play in cells. Study them separately for research. Discover how diseases affect small versus big molecules. Make medicines targeting specific molecules. It's also a basic principle of biochemistry, helping organize the huge variety of chemicals in life. Super simple comparison table. Feature micromolecules, macromolecules, biomacromolecules. Size: small, very large. Molecular weight: less than 1,000 dar, above 10,000, often 100,000 plus. Acid solubility: dissolve in acid, acid soluble. Do not dissolve in acid, acid insoluble. Examples: glucose, amino acids, vitamins, proteins, DNA, RNA, polysaccharides, lipids. Functions: quick energy, building blocks, structures, enzymes, genetic storage. Big picture takeaways. Micromolecules are like single Lego pieces, small, mobile, quickly used. Macromolecules are like huge Lego structures, complex, do specialized, important jobs. Life needs both. Small molecules for energy and reactions, big molecules for building, information, and complex tasks. Knowing the difference helps scientists unlock the secrets of life, from nutrition to disease to biotechnology. Would you like a diagram or a memory trick to easily remember micromolecules versus macromolecules? Certainly. Here's a great, deep, detailed summary of the entire, broken down into the simplest terms. Insoluble and acid-insoluble fractions. When scientists study cells or tissues, they grind them up to break open the cells and separate the chemicals inside. This is done by adding acid to the crushed tissue. The acid causes the smaller molecules to dissolve, while larger molecules stay insoluble, don't dissolve. This gives us 2 main fractions. 1, acid-soluble pool, contains small molecules that dissolve in the acid, includes things like amino acids, sugartides, and water-soluble compounds, represents the cytoplasmic contents, the jelly-like substance inside cells. 2, acid-insoluble fraction, contains larger molecules and things that don't dissolve in acid. includes macromolecules, big molecules like proteins, DNA, RNA, and polysaccharides, and lipid vesicles, lipid structures formed from broken membranes. Represents macromolecular structures in cells and organelles, the nucleus, mitochondria. Hash! Lipids in the acid-insoluble fraction. Why are lipids in the acid-insoluble fraction? Lipids are small molecules, usually under 800 Daltons, so they should dissolve in the acid like other small molecules. However, inside cells, lipids don't exist as individual molecules. They are arranged in structures like cell membranes. When cells are ground up, the membranes break into smaller pieces, forming tiny lipid vesicles, little bubbles made of lipids. These lipid vesicles are too large to dissolve in acid and stay in the acid-insoluble fraction alongside the big macromolecules. Therefore, lipids act like large structures despite being made of small molecules, and they end up in the macromolecular fraction. What are macromolecules and polymers? Macromolecules are large molecules made up of many repeating units. Proteins are made of amino acids. DNA and RNA are made of nucleotides. Polysaccharides are made of sugars. Lipids are not polymers. They don't consist of repeating units, but they form large structures like membranes and vesicles, which behave similarly to macromolecules in this context. Cytoplasm versus organelles. The acid-soluble pool represents small molecules found in the cytoplasm of the cell. The acid-insoluble fraction contains both the macromolecules from the cytoplasm and organelles and lipid vesicles formed from broken membranes. The chemical composition of living tissues. When you combine both fractions, the acid-soluble pool and the acid-insoluble fraction, you get the full chemical composition of a living tissue. Water is the most abundant chemical in living organisms, making up 70 and 90% of the cell's weight. Other chemicals include proteins, nucleic acids, carbohydrates, lipids, and small molecules. Key takeaways. 1. Lipids are small molecules but form large structures, membranes and vesicles. 2. Lipid vesicles don't dissolve in acid, so they end up in the acid-insoluble fraction, just like big macromolecules. 3. Macromolecules are large molecules like proteins, DNA, and sugars that do not dissolve in acid, making them part of the acid-insoluble fraction. 4. Cytoplasm contains the small molecules which go into the acid-soluble pool, while the acid-insoluble fraction includes larger molecules and lipid structures. 5. Water is the most abundant molecule in cells, followed by proteins, lipids, and other biomolecules. In simple terms, when you grind up cells and add acid, you get two parts. Small molecules that dissolve in the acid. These are in the acid-soluble pool. Big molecules, proteins, DNA, etc., and lipid structures that don't dissolve in the acid. These are in the acid-insoluble fraction. Even though lipids are small, they behave like large molecules because they form membranes, which are large structures, so they end up in the acid-insoluble fraction. This entire process is crucial for understanding how cells are structured and how different types of molecules in the cell behave, whether they are small, large, or form complex structures like membranes. Would you like a diagram or some examples to make it even clearer? Of course, here's a great, deep, detailed summary, but still in the simplest language of everything about proteins in your text, expanded so you truly understand what proteins are, how they're built, why they matter, and what they do. What are proteins really? Proteins are big, complex molecules that living things need to live, grow, heal, and function. They are like tiny machines and building blocks inside every cell. Every living organism, from bacteria to humans, depends on proteins for survival. What are proteins made of? Proteins are made of amino acids, which are small molecules sometimes called the building blocks of life. There are 20 different types of amino acids used to make proteins. These 20 amino acids have names like alanine, cysteine, proline, tryptophan, lysine, etc. How are amino acids connected? Amino acids join together one by one to form a long chain. Each amino acid in the chain is connected to the next by a peptide bond, like a special kind of glue. This chain of amino acids is called a polypeptide. Example, think of a protein like a beaded necklace. Each bead equals one amino acid. The thread equals the peptide bonds holding the beads together. Proteins are polymers, but what kind? A polymer is a big molecule made by repeating small parts, monomers. In proteins, monomer equals amino acid. Polymer is polypeptide chain protein. Because proteins are made of different kinds of amino acids, they are heteropolymers, hetero, different. A homopolymer would be made of just one type of monomer repeated over and over, e.g. cellulose is a homopolymer of glucose. Why does having 20 amino acids matter? Having 20 kinds of amino acids lets life create millions of different proteins, each with unique shapes and jobs. It's like having 20 letters in an alphabet to form countless words. The order of amino acids in a protein determines its shape, and the shape determines what the protein does. Essential.
Versus
Non-essential amino acids. Essential amino acids your body cannot make. You must get them from food. Diet. Non-essential amino acids your body can make on its own. Why it matters. If you don't eat enough essential amino acids, your body cannot build certain proteins, leads to poor growth, weak immunity, or other health problems. Dietary proteins. Why we need them. Proteins in our food give us the essential amino acids we need to stay healthy. Foods rich in complete proteins having all essential amino acids include animal sources, eggs, meat, fish, milk, some plant sources, soybeans, quinoa. Many plant foods are incomplete proteins, missing some essential amino acids, but combining them, e.g. rice plus beans, can give a complete protein. What do proteins do in living things? Proteins perform countless functions, including enzymes, proteins that speed up chemical reactions so they happen fast enough to support life. Example, lactase helps you digest milk sugar. Hormones, proteins that send messages in the body and control processes. Example, insulin controls blood sugar. Antibodies, proteins that recognize and attack viruses, bacteria, and other invaders. Transport proteins carry important molecules around the body or across cell membranes. Example, hemoglobin carries oxygen in blood. Structural proteins give strength and support to cells, tissues, and organs. Example, collagen makes skin, bones, and tendons strong. Movement proteins help muscles contract and cells move. Example, actin and myosin in muscle cells. Two examples of proteins. Collagen, the most abundant protein in animals, including humans, found in skin, bones, cartilage, tendons, and ligaments. Gives strength, elasticity, and structure to tissues. Without collagen, our bodies would be fragile and floppy. Rubisco, full name, ribosphate carboxylase oxygenase, the most abundant protein in plants, algae, and some bacteria. Key enzyme for photosynthesis, captures carbon dioxide and helps plants make sugar. Without Rubisco, plants couldn't grow and animals, including us, wouldn't have food or oxygen. Why does protein structure matter? The order of amino acids in a protein decides how it folds into a 3D shape. That shape is critical. It determines what the protein can do. If the order of amino acids changes even by one, the shape can change. The protein might not work, can cause diseases. Example, sickle cell anemia is caused by just one amino acid change in the protein hemoglobin. Big picture summary. Proteins are polypeptides, long chains of amino acids linked by peptide bonds. There are 20 amino acids, making proteins hetero. Proteins are made with specific sequences of amino acids. The sequence gives them their unique shape, and the shape lets them do their special jobs. Some amino acids are essential, must get them through diet. Proteins do nearly everything for life. Build tissues, fight infections, transport molecules, control body processes, speed up chemical reactions. Collagen is the most common animal protein. Bisco is the most common protein in the entire biosphere. Why this is important for you? Understanding proteins helps explain how your body grows, heals, and functions. Shows why a balanced diet with enough protein is vital for health. Explains diseases that happen when proteins are missing, misfolded, or not working. Would you like a chart of the 20 amino acids or a step-by-step look at how proteins fold into their final shape? Of course, here's your great, deepest, detailed summary of everything you just read about polysaccharides, but still in the simplest possible language with every important point expanded so you can truly understand and remember it. What are polysaccharides? Polysaccharides are very large molecules, macromolecules, made by linking many small sugar units, monosaccharides, together. The word poly means many, and saccharide means sugar, so polysaccharides literally mean many sugars. They can have hundreds or thousands of sugars in one long chain. Polysaccharides are a type of carbohydrate, just like simple sugars, but much bigger. How are they built? Polysaccharides are like a necklace made of beads. Each bead equals one monosaccharide, like glucose or fructose. The string equals bonds holding them together, called glycosidic bonds. The necklace can be straight, linear, or branched. The chains can be homopolymers, made of only one type of sugar, repeated over and over, e.g. cellulose only glucose. Heteropolymers, made of two or more different sugars, not common among the basic examples here. Examples of important polysaccharides. Cellulose, made only of glucose repeated many times, so it's a homopolymer. Chains are straight, rigid, and packed tightly together, found in plant cell walls, giving plants their strength and stiffness. Humans cannot digest cellulose, it passes through as fiber. Starch, also made of glucose, but chains are helical, spiral-shaped, and branched, found in plants, used to store energy in roots, seeds, and tubers, potatoes, rice, wheat. When we eat starch, our body breaks it into glucose for energy. Iodine test. Iodine fits inside starch helices, turns blue, shows starch is present. Glycogen, similar to starch but found in animals, including humans. Chains are highly branched, allowing for quick release of glucose when we need energy fast. Stored in liver and muscles, our emergency sugar battery. Inulin, made of fructose, repeated homopolymer, found in plants like onions, garlic, chicory. Acts as dietary fiber for humans, feeds good bacteria in our intestines. Chitin, a tough polysaccharide made of modified sugars like N-acetylglucosamine, found in exoskeletons of insects, spiders, crabbers, makes their shells hard and protective, like armor. How do reducing and non-reducing ends work? Chain has two ends. The reducing end can react chemically, e.g. donate electrons. The non-reducing end, more stable, doesn't react easily. Enzymes that break down polysaccharides often start chewing from the non-reducing ends, like eating from the outside of a candy rope. Why starch turns blue with iodine but cellulose doesn't? Starch chains twist into helices, spirals. These spirals create spaces inside the chain. Iodine molecules fit into these spaces, and when they do, the solution turns blue. That's the iodine test for starch. Cellulose doesn't have helices, its chains are straight and tightly packed. No space for iodine, it doesn't turn blue. What makes cellulose strong? In cellulose, straight chains of glucose line up side by side and hydrogen bonds form between chains. These bonds act like glue, making cellulose very tough and rigid, perfect for building plant stems, wood, and fibers. What are modified sugars? Some polysaccharides sugars, but also modified sugar, like amino sugars, sugars with nitrogen atoms, e.g. glucosamine and chitin. Chemically modified sugars, sugars with extra chemical groups, e.g. N-acetylgalactosamine. These special sugars give polysaccharides new properties: strength, flexibility, or resistance to decay. Where do we see polysaccharides in daily life? Paper and cotton, made of cellulose from plants. Food like bread, rice, potatoes, starch gives energy. Animal livers store glycogen for quick energy. Insect shells, chitin, makes them hard and crunchy. Dietary fiber, cellulose and inulin, keep digestion healthy. Big picture, why are polysaccharides important? They are key structural materials, e.g. cellulose in plants, chitin in animals. They are main energy stores, e.g. starch in plants, glycogen in animals. They provide fiber for digestion, cellulose, inulin. They are involved in biological protection, e.g. chitin makes insect exoskeletons. Deepest takeaway summary: polysaccharides are giant sugar chains that can be straight or branched, made of the same sugar, homopolymer, or sometimes different sugars. The way sugars connect and the type of sugar used give each polysaccharide its such special role: strength, energy storage, or protection. They are essential to plants, animals, humans, and the whole ecosystem, from building plant structures to storing energy in our bodies. Would you like a diagram of cellulose, starch, and glycogen structures, or a memory trick to remember their differences?